Hello, in this video we are going to talk about the history of microbiology, scientists' names and their discoveries, important landmarks and the vaccines described by the important scientists. So what are those discoveries? This is very important topic with respect to the various exams like GATE, Life Sciences, CSIR, UGC, NET, and of course if you are in 11th or 12th class, for such situations this is a very important topic. Antony van Leeuwenhoek, he was the father of microbiology. He is credited for firstly observing the bacteria. He had an hobby of grinding lenses and observing diverse materials through them. So he observed bacteria and other microorganisms using a single lens microscope of his own design. So what Antony did, he used to make the simple microscopes of his own design. It was just grinding lenses, and so that he can see the small objects, small microorganisms on a large scale of big to make it magnify. So this is what he did. Louis Pasteur. Louis Pasteur is credited with a lot of inventions and techniques. He is also named as the father of microbiology. Antony van Leeuwenhoek is also named as the father of microbiology. He is credited with the sterilization techniques, and he also developed the stream sterilizer, hot air oven, and autoclave. He also established different growth needs of different bacteria and contributed to the knowledge of anthrax, chicken cholera, and hydrophobia. He attenuated cultures of the anthrax bacillus by incubation at high temperature and proved that inoculation of such culture in animals induced a specific protection against anthrax. So what does it mean? It means he discovered that when a microorganism which is viral, which induces viral symptoms in other organisms, if we induce a small portion of it in the body, for example, like we have COVID vaccine. So what is happening, we are basically inoculated with a bit of COVID vaccine protein structure so that antibodies are made in our system. So this is basically what was the approach of Louis Pasteur. What he discovered, that if we inoculate a small portion of viral organism, then it can induce antibodies in our system. Now let's move to the next point. It was the Pasteur who coined the term vaccine. So vaccine term was coined by Louis Pasteur for such prophylactic preparations to commemorate the first of such preparations, namely cowpox, employed by Edward Jenner for protection against smallpox. So what Edward Jenner did, he devised vaccine against smallpox, and what we will say, such kind of medicines. So it was Louis Pasteur who devised the term vaccine for such kind of medicines. You can say it as medicine, but it is not. It is vaccine for such kind of preparations. It is not, you can say it as a medicine or not. The greatest impact on medicine was made by Pasteur's development of a vaccine for hydrophobia. If we know that what is hydrophobia, it is a kind of fear, so against water. So he devised a medicine against the hydrophobia. Now Joseph Lister affecting a pronounced. So he used antiseptic surgery involving the use of carbolic acid. So carbolic acid is an antiseptic. So Joseph Lister devised this technique during the surgery, which did help in mortality rate increase. For example, during surgery many people died because of poor sanitation conditions, but absence of aseptic conditions. So what he did, he used carbonic acid for these purposes. Robert Koch in Germany perfected bacteriological techniques during his studies on the culture and the life cycle of the anthrax bacillus. He studied the on and life cycle of anthrax bacillus, like how it grows and reproduces, all these things. He invented bacterial culture method. And so this is a very important point. You can ask that who devised the pure culture technique using we for example, in microbiology, what happens, there are agar plates on which there are medias, and we grow bacteria on it. So this technique was devised by Robert Koch. This technique of culture was devised by Robert Koch using the media. He invented bacterial culture method. He introduced staining techniques and methods of obtaining bacteria in pure culture using solid media. So what he devised, the solid media and the agar plates, and on which you can grow the bacteria. So he did that. He discovered the bacillus of tuberculosis, anthrax, and the cholera vibrio. So what it means that there are, at that time there were diseases like TB, vibrio, anthrax. So what was the cause of these diseases? He discovered these bacteria responsible for these diseases, which were TB, anthrax, and the cholera vibrio. Rooks and Yersin. Rooks and Yersin identified a new mechanism of pathogenesis when they discovered the diphtheria toxin. Similar toxins were identified in tetanus and some other bacteria. The toxins were found to be especially neutralized by their antitoxins. So what happens that if there are bacteria, what happens if they enter in our system? Why people get killed because of diseases, so, or they get ill because of diseases? So they discovered the reason that it was a toxin. So when they were working on a particular kind of disease, they found that there is a toxin named as diphtheria toxins, and these toxins were found to be neutralized by antitoxins. Ernest Ruska developed the electron microscope, enabling the visualization of the microbes we now call as viruses. Koch postulates the causative agents of the various infectious diseases were being reported by different investigations in such profusion that it became necessary to introduce criteria for proving the claims that a microorganism isolated from a disease was indeed causally related to it. Now, what does this means? It means that at that time there were happening a lot of discoveries, a lot of microorganisms were get found. So so it was there was a confusion in the community, scientists, doctors community, that whether for example, if there is a disease, is the microorganism found and are claimed to be reason for that disease is actually the reason for that disease? So for these things the Koch postulates were formed. So there were some postulates were made, and if these criteria were satisfied, then they were said that yes, the microorganism that is claimed to be the reason for that particular disease is the really the reason for that disease. So for satisfying that claim, they have to satisfy the Koch's postulate. Alexander Fleming made the accidental discovery that the fungus penicillium produces a substance that destroys staphylococci. Work on this at Oxford by Florey Chain and their team during the Second World War led to the isolation of the active substance penicillin and its subsequent mass production. This was the beginning of antibiotic era. What this means? This means that during the Second World War, Alexander Fleming, who was a scientist, was working on a kind of fungus called penicillium, and he found that this fungus actually destroys staphylococci. So this led to the discovery of the antibiotic, and it helped during the Second World War. But eventually scientists get the result that the bacteria is developing a kind of immunity against the antibiotic. So work on this at Oxford by Florey Chain and their team during the Second World War led to the isolation of the active substance penicillin and its subsequent mass production. Ignaz Semmelweis in Vienna. In fact, even before the microbial cause of infections has been established, Ignaz Semmelweis in Vienna had independently concluded that puerperal sepsis was contagious. Semmelweis also identified its mode of transmission by doctors and medical students attending on women in labor in the hospital, and had prevented by the simple measure of washing his hands in an antiseptic solution, for which service to medicine and humanity he was persecuted by medical orthodoxy and driven insane. Forgive me for my pronunciation if I'm wrong somewhere in pronouncing the names, like I'm not good at pronouncing the names. These slides are very important for you. Now you must know a few more names like Robert Hooke. Robert Hooke is credited with visualizing the first person who saw a microorganism with a microscope was Robert Hooke. Yes, Anthony van Leeuwenhoek saw the microorganism, but it was not, he saw it through lenses which he just experimented with, and to magnify the small microorganisms. But actually the person who first saw the microorganisms with an actual microscope was Robert Hooke. Edward Jenner. Edward Jenner was an English physician, and he is pioneered with the concept of vaccines, including the creating the smallpox vaccine, the world's first vaccine. So he created the first vaccine, smallpox vaccine, and the term vaccine was coined by Louis Pasteur. Are the Koch postulates. According to these, microorganisms can be accepted as the causative agent of an infectious disease only if the following conditions are satisfied. The bacterium should be constantly associated with the lesions of the disease. It should be possible to isolate the bacterium in pure culture from the lesions. Inoculation of such pure culture into suitable laboratory animals should reproduce lesions of the disease. It should be possible to re-isolate the bacterium in pure culture from the lesions produced in experimental animals. An additional criteria introduced subsequently was that bacterium should demonstrable in the serum of patients suffering from the disease. So these are the Koch's postulates. Theory names you should know. There are some names you will encounter in the microbiology history. These are the theory of spontaneous generation, which was disapproved by Louis Pasteur. मतलब कि it means that Louis Pasteur said that it is wrong theory. Spontaneous generation, theory of spontaneous generation is wrong. Next theory you must know about is germ theory of disease, and third theory is very important, which is Koch's postulate. So you must know about these theories. These are some of the important topics. I hope this video helped you, even if my pronunciation was wrong. The things are written here are facts. You can refer to these slides for your help, for information. If you're unable to understand what I'm trying to say, then you can just go through the slide, read it, what the content is written it, and just you can go through the Wikipedia page. Just search in the Google bar the history of microbiology, and you will a good info in the history of microbiology corner. Just scroll through and check the history of microbiology corner. There is a good info there. And please don't try to make notes, too much notes. It is not beneficial. Marking notes is not beneficial. Just go through it and just try to revise as much as you can. It is the thing, it can help you, and revise it.